Thanks, Chris, for that great introduction. So I'm Geoff Meacham. I'm the Senior Biotech and Pharma Analyst, and we have Dan Skovronsky with us from Eli Lilly. So welcome, Dan. Looking forward to this chat here. So I guess maybe let's just start off with the news of the day or from last week. Give us some context for the really remarkable data that you guys showed for tirzepatide in obesity, and maybe help us with how that could potentially be paradigm shifting moving forward.
Yeah, thank you, Geoff. Thanks for having me here. Thanks to the organizers. Great to be together today. It's been an exciting couple days for us. We just top-line data from our molecule called tirzepatide. This is a dual-acting incretin, two different incretins combined in one molecule, GIP and GLP-1. And with it, we saw what is, I think, the largest degree of weight loss by some margin that's ever been demonstrated in a large clinical trial in obesity. A 22.5% body weight reduction at the highest dose on average, with many patients achieving more than that. That equated to a 52-pound weight loss by the end of this trial. So we see that as a life-changing amount of weight loss, likely to be hugely medically important. Over time, we'll collect outcome data. I'm sure we can talk about that, Geoff. But we know from other studies, primarily around bariatric surgery, that when you lose that amount of weight and fundamentally correct obesity in a majority of patients, you reduce the risk of morbidity and, importantly, mortality from heart disease, from stroke. You have positive effects on joint health, lower risk of cancer, of infectious disease. So the long-term benefits of this, which are yet to be determined, could just be incredible. We're so excited about what we're starting to do here.
And before we get into the more theme of this conference, gene and cell therapy and nucleic acid, just wanted to follow up just on your last comment. So thinking about kind of what are the commercial hurdles to make a drug like tirzepatide in obesity broadly available. You have obviously a pretty restrictive payer policy going back several decades, but that could change with the advent of newer therapies, obviously.
Yeah, it's an important comment, and unfortunately, obesity as a disease has been sort of underinvested in, undertreated. And there are probably significant societal biases and impediments to patients getting treated with newly available therapies. In the past, there haven't been great drugs for obesity until quite recently, so it's not fully surprising. I think there's still a lot of people out there, even a lot of physicians, who might say, obesity isn't really a disease, maybe it's a choice. Exercise more, diet more, and you can fix this. That's really not true. And it was pretty striking, for example, in our study, which is consistent with past studies in terms of the placebo group. These are patients that are highly motivated. They want to get treatment for their obesity. They enroll in a randomized placebo-controlled trial. They're under the care of physicians. They're given a caloric-restricted diet. They're given instruction, encouragement to exercise with follow-up visits to doctor. The placebo arm is like taking a weekly injection of placebo, which placebo works in many different disease states. So it's your optimal setup for weight loss through behavioral modification in the optimal patient population. What did we see in that placebo group? 25% of people just quit the placebo group because they couldn't, they were unsatisfied by what was happening. And of the rest that stayed in the trial, the people who actually stuck with the diet and exercise, about 2.5% weight loss. So when we think that people can modify their obesity through lifestyle, it's a pretty small effect, 2.5%. What we can do with a medicine like tirzepatide, 22.5%. So a whopping difference there. It's not too many clinical trials where you see a tenfold difference between drug and placebo like this. I think, yes, there will be obstacles with payers eventually. Our first launch for tirzepatide, of course, will be in type 2 diabetes, a separate indication, a separate clinical trial program that's under review at the FDA. We hope to bring that forward to patients sooner. But then when we come to the obesity market, it'll be an opportunity to educate doctors and payers about the benefits. So we have a pretty big investment program here, not just to demonstrate the degrees of weight reduction that we can get in patients, but also importantly to show some of the outcomes that come from that. So one of the first things we're working on is outcomes from HFpEF, heart failure with preserved ejection fraction. There we think weight reduction could have a significant impact in a relatively short amount of time on those patients. We're working in sleep apnea, another serious disease where in people with obesity and sleep apnea, weight reduction is quite likely to improve that condition. And we're launching a large morbidity and mortality outcome study that's going to measure a number of outcomes, of course, the usual cardiovascular outcomes, but other health outcomes as well that are implicated by obesity. So over time, we'll be building this wealth of data. So will our competitor in this space, Novo. And I think together we probably have a job to do to educate doctors and payers about the importance and the economic benefits even for payers of having patients lose weight and reduce other medical conditions.
Well, Dan, let's shift gears to go from protein-based therapies to nucleic acid, and maybe just remind us of what Lilly's capabilities are with respect to nucleic acid therapies. I know you have a couple of mRNA platforms, but you're not one of the leaders yet, at least in gene and cell therapy, and that could be obviously something going forward.
Yeah, it's an interesting area for us. I think at Lilly, of course, we're interested in rare diseases to some extent, but as one of the largest pharmaceutical companies in the world, we also feel a sense of obligation to work on the major medical problems that affect most people. And the highly prevalent chronic diseases are tough targets for gene and cell therapy. I think the challenge that we take on is how to translate the remarkable advances we've seen in relatively small diseases, genetically defined, and move them into bigger spaces. So one of the places we've chosen to work is CNS. Another is cardiovascular disease. I probably just quickly touch on our efforts in those areas. In CNS right now, we have two major areas for investment. In gene therapy, we were fortunate to be able to acquire Prevail Therapeutics based in New York, working on AAV directly injected in the brain, working on neurodegenerative diseases, initially starting out with genetically defined neurodegenerative diseases in the patients who have a specific deficiency, a GBA1 or progranulin, our first programs, to correct that with AAV, transduced gene therapy, and see if we can impact those diseases with a hope of eventually broadening to other populations. Huge challenges there in route of administration, of course, in transduction, how many neurons and what part of the brain can we reach, and in timing of therapy. But we're really excited to be working with that team, and those molecules those programs are in phase 2 for their respective indications. The other approach we have is siRNA, so trying to turn off genes, the opposite of our approach with AAV, where largely we're turning on genes or replacing defective genes. So we're turning off genes with siRNA. And here we have built a lot of internal capabilities as well as some partnered capabilities, and focus in CNS initially on intrathecal delivery through lumbar puncture, but ultimately we'll grow to intravenous administration if we're successful. And what we've been doing here is modifying siRNA by conjugating it to small molecules, lipids, peptides, proteins, antibodies, to try and improve its ability to enter neurons and turn off genes. We're having some pretty good success. We can now, after single lumbar spinal injection, knock down gene expression throughout the brain of a primate to pretty significant degrees. So we're excited about that and moving those programs into humans in coming years. Of course, like many other companies, we've also worked in cardiovascular disease. There it's a bit easier because we're targeting the liver, readily available for gene and cell therapy, in this case with siRNA, working on targets like Lp(a) and ANGPTL3, which of course are risk factors for major causes of cardiovascular disease.
Well, Dan, to follow up on that, when you talk about to tie it together, scaling up to larger indications, like say obesity or diabetes or Alzheimer's, and then having proof of concept for a nucleic acid derived therapy, how do you marry those two together after you've had a de-risking event? How do you invest in a scale up piece of it, after your initial proof of concept, and how does Lilly think about scaling up a technology like that for a big indication?
Yeah, so we actually just start with that end in mind. So in other words, we're happy to pursue and proud to pursue small disease areas, orphans, genetically validated diseases, if we see that road to stepping up to a bigger disease. So it probably doesn't make sense for us to devote a significant fraction of our resources to something that will literally help thousands of people if there's no path from what we learn there to helping bigger and bigger populations. But fortunately, usually that's not the case. Usually we do see a path where we can de-risk a technology. We can learn more about targeting AAV to neurons in the brain, or we can learn more about getting siRNA to enter neurons. And then we can learn that while solving a problem in a smaller genetically defined disease. But then once we've cracked the code, and in this case I'm focusing on CNS delivery, once we've cracked the code on CNS delivery, whether that's virus or siRNA or nanoparticles or anything else, we can broaden with different payloads to other diseases. I think that's really the power of genetic medicines. It's not like a small molecule drug discovery program where each one is its own work of art and a bespoke project. Our hope is that in this area it can be more like a platform where once we've solved some of the fundamental platform challenges, we can apply it across multiple diseases and multiple targets.
And Dan, just on the CNS front, you guys obviously have had some de-risking data so far for donanemab. Just maybe walk us through kind of how Lilly's thinking about this going into some of your data coming up, the head-to-head study on PET scan, but broadly the indication, and what CMS and that whole controversy with respect to Aduhelm kind of did to Lilly's strategy.
Thanks, Geoff. It's an exciting time in Alzheimer's disease for sure. I've been working in this area for more than 20 years, and I think we've never felt more excited about the future, really based on our donanemab phase 2 Trailblazer data, which still today, I think, despite all the controversy and discussion in the field, in Alzheimer's disease, we have one positive randomized controlled trial for a disease-modifying drug, and it's that donanemab trial. Everything else has been a near miss, or look at the subgroup or post-hoc analysis. So we have a firm foundation here in a positive phase 2 study. We're replicating that now in a phase 3 study. We'll get those results the middle of next year, which is just a blink of an eye away. So really excited to see that. There's some unique attributes with donanemab that I think give us more confidence it'll be successful. I see in general that amyloid-lowering drugs are likely to have a positive effect, although those effects could vary. They could be on the smaller side. There could be a lot of noise in clinical trials. So we've done everything we can to optimize the benefit here for patients. That means having a drug that's extremely potent, can clear plaques quickly, because in an 18-month trial, if you're only clearing plaques at the end of the 18 months, there's not much time for a downstream benefit in slowing cognitive decline. So we need to get those plaques out of there in the first 6 months at least of the trial. And then you need to treat the right patients. And Lilly's been a leader in that area. We brought forward amyloid imaging, which now everyone uses. Now we're doing tau imaging, which is a great way of stratifying patients for therapy. We've brought forward a phospho-tau blood assay, which looks extremely promising for identifying patients for therapy. So getting the right patients in your trial, treating with a potent drug. And then the third problem we have in Alzheimer's disease is outcomes. And all of the outcomes we have, essentially are giving cognitive tests or exams to patients, or asking their caregivers how they're doing. Those kinds of things are pretty noisy. We've invested a lot in finding the most reproducible, least variable outcome measures that are still meaningful to doctors and patient caregivers. And so we've designed our trial to use those outcomes as well. A lot of controversy here. We've done things differently than other companies. I think that's right. We're advancing the field here. But I think if we have a positive phase 3 study next year on the heels of a positive phase 2 study, that will be a compelling body of evidence. I'm surely disappointed that CMS has chosen to sort of in advance proclaim a lack of coverage for drugs that are FDA approved for Alzheimer's disease. That makes no sense to me. On the other hand, I think when we have that compelling evidence, we'll be in a good position to reopen that discussion and change the policy. I don't see any world in which we have an Alzheimer's drug that is a clean success, that FDA fully approves, that thought leaders and scientists around the world recognize as a benefit for patients, and Americans can't get it because CMS isn't going to pay for it. That's not going to happen. So we see this as potentially delays and frustrating for us and for patients most of all. But in the long term, we'll fix it.
Let's talk a little bit about the why you've been successful. It's about the molecule in Alzheimer's. It's also about the imaging piece, though, and the patient stratification. And maybe just talk a little bit about kind of the role of PET imaging and tau imaging, sort of live to help, stratify patients and to increase your chance of success.
Yeah, thanks, Geoff. And this is a huge theme, I think, in neuroscience, but really in every difficult disease that we work on. It's identifying the right patients. And so many, particularly highly prevalent diseases, are massively heterogeneous in the populations that we seek to treat. And that serves to do two things. One, introduces noise in your clinical trial because a heterogeneous population, some people are progressing, some people don't have the disease at all, and they just get better naturally. So you have a noisy placebo arm. The other is that you'll have patient groups that don't respond to therapy if they don't have the disease of interest. So the first observation we made after a decade of investing in biomarkers was that 5 or 10 years ago, a third of the patients people were enrolling in Alzheimer's trials didn't even have Alzheimer's disease. They didn't have amyloid plaque in their brain. So we had developed this amyloid imaging agent essentially for this purpose. That was the first FDA-approved amyloid imaging agent, and now widely used in clinical trials. No one does anti-amyloid trials in people who don't have amyloid in their brain. That's nonsensical. The second advance we brought forward was the tau imaging. And it turns out even if you have people with clinical stage Alzheimer's disease who have amyloid plaque in their brain confirmed by imaging, some of them might have no tau pathology in their brain at all. That's a very early stage of the disease. And over 18 months, we've seen in previous studies that those patients actually don't progress at all on placebo. So it's not a good clinical trial patient population because in the lack of progression, there's no ability to show a drug effect. People with more tau progress faster. So if you're sort of really want to power your study and greedy for effect size, you might say, hey, let's go after the people with a lot of tau. We know they go downhill fast. It should be easy to show a drug effect. That might be good for some mechanisms. I think for amyloid-targeted mechanisms, it may unfortunately be too late for those patients. So we excluded both ends of the tau spectrum, focused on those intermediate tau, about 2/3 of the amyloid-positive population, who we think will progress at a predictable rate and have the best opportunity for a drug effect with anti-amyloid. Competitors have chosen different routes. It's going to be an exciting 12 months here as we see readouts from 2 other companies and then ours. And then we can compare across these different strategies and see how much some of these biomarkers might impact our ability to see efficacy.
And let's switch gears. There's a lot to talk about in your pipeline, but you have a really robust oncology pipeline. Verzenio and early stage breast cancer is just now approved and launching. But pirtobrutinib, BTK inhibitor, is also advancing in the clinical studies. Help us with kind of how Lilly thinks about oncology as kind of a growth pillar going forward, and what are some of the more exciting things that you could add to combination therapies or other indications that you're looking at that are compelling?
Yeah, thanks, Geoff. Oncology is certainly the most competitive area in which we work. Everybody is working hard to solve problems for people with cancer. That's good news for patients. Makes our jobs a little bit harder, but we relish that challenge. Verzenio is probably a good example of that. many years ago, we worked to develop the CDK4/6 inhibitor. We thought it had the potential to be the best molecule based on some of the preclinical attributes, but we were third coming to market. But still, we thought we had a differentiated molecule. We invested in differentiated clinical trials. And over the last many years, we started to accumulate data that this molecule is different. We had monotherapy data. We had the OS data in metastatic breast cancer. And then last year, we had the success in adjuvant breast cancer, where we stand alone in the CDK4/6 class. So really excited to have launched that indication recently for a subset of women with breast cancer in the adjuvant setting, seeing great uptake with physicians. And now finally, after many years of investment, we see Verzenio becoming the NBRX leader in the CDK4/6 class. So that's exciting, big opportunity to help women. And then now we also have an ongoing study in prostate cancer that progressed to phase 3 based on a blinded analysis of phase 2 data that met a pre-specified threshold for progression. Prostate cancer is sort of a mirror image in many ways of breast cancer, and hopeful to see great efficacy in phase 3. Pirtobrutinib is another sort of neat story in oncology, designed initially to overcome the C41S mutation in BTK, a mechanism of resistance to conventional BTK inhibitors. So we made this molecule thinking this would be a highly targeted therapy for patients with that mutation, but quickly discovered that it's actually just a great BTK inhibitor. And patients who fail first-line BTK inhibition in CLL and MCL and other B-cell lymphomas actually have a pretty high response rate to this novel BTK inhibitor, pirtobrutinib. So excited about that moving forward in a number of settings, initially the second-line settings and then ultimately first-line. First submission underway here for MCL in the second-line setting and then grow from there. I see this as a big opportunity for patients. We'll continue on those themes that I outlined. So making molecules that are best in class, that are exquisitely tuned for patients to deliver differentiated efficacy in a safe way, fully engage highly validated targets where we know if we hit the target, we can have a big effect. Molecules like our KRAS inhibitors coming behind that, our PI3 kinase alpha mutant specific inhibitor that'll be entering human trials this year, match that paradigm as well. So really excited about that future in oncology.
So a lot of these indications in oncology, hematology, are monotherapy, but going forward though, help us with maybe the potential to, for combination therapy, maybe to overcome resistance and the like, but also the potential to involve other modalities such as gene therapy or cellular therapy or any sort of genetic medicine as a combination as well.
Yeah, there's no doubt that combination therapy is the mainstay probably of treatment of most cancers. I think we hold a pretty high bar. By and large, we expect to see single agent activity in phase 1 for a cancer agent if we're to proceed. I know others take different strategies. There's been a lot of investment sort of in empiric drug development, looking at combinations of different things across different diseases, hoping to see differentiated activity. I think that's too hard, time-consuming, resource-intensive for us right now. So we're looking for drugs that have monotherapy, but then combine them. There are many challenges there. So for example, in KRAS G12C inhibitors, can you combine it with a PD-1? That would be great. So we work on things like that. For CDK4/6, a SERD inhibitor, an oral SERD. So we work on that. For BTK, BCL2 is an obvious partner there. We work on our own BCL2 inhibitor as well as the commercially available one. So I think those will continue to be important for patients, but we probably need some clinical de-risking earlier. In terms of gene and cell therapy, we frankly haven't invested yet in oncology. I think for a number of reasons, we see that a little bit less compelling than CNS, where we have sort of unique opportunities in the brain as a protected space, as well as sort of systemic diseases where we can target liver or blood.